Here, in Yellowstone, are a type of bacteria evolved to live under very specific conditions. The springs they live in get to 92 degrees Celsius, almost boiling, with steam vents reaching temperatures of 135 degrees. These conditions are too hot for most species on Earth, but the TAC bacteria here are able to perform basic cellular function, specifically the replication of DNA. To do this, they use the highly specialized enzyme called TAC polymerase, which is the key to efficient DNA replication and testing. PCR is not a type of test itself. Instead, it is a process in which DNA can be amplified. When retrieving a sample, it is possible the amount of DNA is extremely small, so through PCR, the amount of DNA can be replicated and increased by a large factor. The first step in PCR is to obtain sample DNA, the most easily identifiable example being a nasal swab. It is then placed in a buffer solution with a pH between 8 and 9.5, including other salts. Heating to 90 to 100 degrees Celsius to split the DNA, this is called denaturation. One of the salts included is KCl, which forms K plus ions that help with the denaturation stage next. This stage is called annealing and is when primers or small complementary strands bind to their respective sections. The primers must anneal in order to define where the DNA should be replicated. For the annealing process, the temperature must be decreased to 30 to 65 degrees Celsius. The final step is extension, when DNA is heated to 60 to 75 degrees and the Taq polymerase makes a new second strand. The result is two strands of DNA, one new half and one old half.